characteristics of biodata in terms of the general ideas of big data today. Life sciences data have very little structure. They are getting larger very rapidly, and they have very high heterogeneity. Many, many different data types coming from many different instrumentation types, and the types and the technologies are very unstable. They're changing on a very rapid basis. So we have many different file formats that we have to cope with, wildly different sizes, differing acquisition rates, and there's still considerable manual data collection. A lot of very important information hides in people's notebooks, and if we're trying to integrate large data sets together, we've got to be able to track that information as well, otherwise we lose information. We have a huge range of different analysis methods, both computationally, statistically, and methodologically, and they change rapidly too. So we have many different metadata standards in the life sciences, which is in some ways good and in some ways adds to the complexity of what we try to do when we store and use and reuse data meaningfully. And we have an additional issue of confidential patient data, private data, that increasingly has to be integrated at different levels with pure research data in order to actually make sense of the data sets that we are generating. So what sort of data are we talking about? What do I mean by data? Well, sometimes we're talking about raw data files, sometimes we're talking about analysed data files, sometimes results, which can go across many different data types, many different files. We need to store hypotheses, and hypotheses are meaningless without the context in which they're formed. We often have to store standard operating procedures. Sadly, many of our researchers don't like generating them, but they're very useful when you want to share and reuse data meaningfully. And we also generate software, as I'm sure most other people do as well. We generate tools and we generate interfaces. Unfortunately, many researchers still don't believe that those are data assets themselves, so they sometimes get a bit lost. And we have a large amount of dark data. So what's dark data? So it's miscellaneous additional data that isn't necessarily associated with any particular publication, but is still meaningful and reusable. And we need to not lose track of that while we go. In terms of biological data sets, these are a few of the different types of data sets that are generated in any particular research lab. One experiment may generate many of these different data sets, or it may only generate one, that each have their own standards, each have their own formats, are associated with particular different instrumentation types. And we need to be able to integrate those data together to form meaningful analyses more and more, and that in itself is a major hurdle. I managed to generate about 1,000 bases of sequence during my entire PhD project. Nowadays you can sequence the entire genetic complement of a human in 27 hours, and it will cost about a few $1,000s. So we have to consider very carefully what we're going to do with all of this data. And it also needs to be associated with sufficient experimental data that we can actually use it meaningfully with other data types as well. So are we really producing big data? Well, this paper at the top is a very interesting one. There have been some basic comparisons between how quickly we are able to generate just DNA sequence information, its volume, and the projections on the instrumentation we have now. Where are we going to be in terms of volume in the next 5 years? At the moment, if we just looked at all of the DNA sequences that we know have been sold and that are currently functioning, we should be able to generate about 35 petabases of DNA sequence per year. And that's without technology getting faster. So we have a big data problem here. Even one small lab can generate an awful lot of information. So sequencing 170 human genomes is the sort of thing that many labs now do on a fairly regular basis. And just that one experiment is able to produce 40, 50 gigs of the publication level data, the sort of data that you need to send to public repositories. And if you need to store the raw data from that sort of size project is a few terabytes, in fact. So that mounts up awfully quickly. And if you're going to analyse those data and keep the appropriate metadata to tell you how those data were generated and what to do with them, That's quite a big challenge in itself because these file formats are not particularly standardised, they change over time. Different software brings in new versions, so if you want to be interoperable with data you generated 3 years ago, that can be quite a computational challenge. Another big problem in the life sciences is we've been very good at publishing data for a very long time, and that's led to a huge amount of fragmentation. On top of that, we have the clinical data problem. Some of the data we generate, even if we want to make it publicly available, can never be openly published. So data that is identifiable human data has to be handled separately, but we need to be very careful to decide what we keep and what we throw away, what is reusable and what is not. There are holes in how data are managed. We also use basic laboratory notebooks to try and encourage researchers to capture data and back it up rather than just write it into paper notebooks. And we work closely in trying to make reusable pipelines to encourage better capture of metadata.